Hello, my name is Shalini Lonam. I am an assistant professor of chemistry and physics here at Purdue University, and I definitely want to start by thanking the organizers for the invitation to speak and also all of the coordination efforts that have gone into making this all possible so that even in a virtual context we can still have great interactions. So today I'll be talking about work from largely my group here at Purdue, but also some work from my postdoc at UC Berkeley on the topic of understanding T-cell activation by using single molecule methods. Okay, so I'll start by playing this video acquired by my student Kevin that shows the remarkable behaviors of chimeric antigen receptor T-cells, in this case primary CARs, that are sampling a breast cancer cell. And you can see that these spherical cells come and interact with this large flat cell. And in fact, quite remarkably, in the case of the main activity you can see here, this T cell actually burrows underneath the tumor cell. And it clearly shows that there can be some robust interactions between these two different cell types, and we know that the development of CARs has been an exciting new area of treatment option for certain cancers that may give robust killing, given that they can be developed from one's own immune system. So I'll start by acknowledging my graduate student, Kevin, who has been just absolutely fantastic over the last couple of years, and he certainly represents the greatest accomplishment in building my lab since I started here at Purdue in 2019. He is an incredibly industrious individual and has just a great spirit for imaging and for collaborative work. So his accomplishments are really fantastic, and I'm grateful for everything he contributes. Our lab does work in the area of biochemistry and biophysics, and in particular focuses at interactions at cell-cell junctions, because a lot of important reactions occur at these membrane-membrane interfaces, and the membrane itself serves as a reaction landscape. It's also important to remember that very often the critical activities that are happening at these interfacial spaces involve only small numbers of molecules. And that's not just the ligands and receptors that may be interacting, but even the downstream signaling molecules. It may in fact be the activities of very small numbers of molecules that are actually most influential in the overall behavior. And furthermore, that you can get heterogeneous outcomes from genetically identical cells, which is to say that even as we start to develop these therapeutics based on chimeric antigen receptor T cells, it could in fact be the case that the activities of small numbers of cells actually dominate the overall outcome of the system. The mechanics are also really important in these spaces where these tortured membrane interfaces and the stiffness of the local environment can really impact the success of signaling. And so in trying to think about all of these themes, we start with a beautiful example, which is the T cell itself. And during my postdoc at UC Berkeley with Jay Groves, we really focused on T cells, which are fundamentally single molecule detectors. And so what you see here is a depiction of the interface between an antigen presenting cell, or APC, and a living T cell. And importantly, the activating information that is transmitted from one to the other occurs at this junction between the T cell receptor, which is shown in purple, and its ligand, peptide MHC. So peptides from antigenic molecules that are presented on major histocompatibility complexes. And just single binding events between TCRs and pMHCs are sufficient to kick off signaling within the T cell that lead to decision-making outcomes. But remarkably, these single binding events must create robust signaling in the context or background of cellular noise and ensure that the T cell can quickly discriminate and act on small numbers of activating events, because of course a T cell mostly encounters in its native context self-antigens and yet must respond to foreign antigens very, very sensitively. And given that they are these beautiful single molecule detectors, one might wonder how we try to get information across a population of cells. And so the importance of understanding single cell can be underscored by thinking about an experiment like flow cytometry, where we can quickly get lots of information about, say, tens of thousands or maybe even millions of cells, but of course we only have a snapshot from an instant in time. And so we would argue that there's a lot of additional information that is in the underlying kinetic that we can better appreciate by gaining additional dimensionality to the data at the single cell and single molecule level, But in the particular case of a T cell, single molecule is additionally important given what I just mentioned about this initiation of signal by small numbers of ligands, even just a ligand. So here is a classical example from the Davis lab at Stanford, where you can see that a single pMHC, which is this bright red dot that you can see, is actually sufficient over several binding events with a living T cell to actually produce the cytokine IL-2. And so this idea of single molecule detection within a population of cells and the possibility for heterogeneous outcomes really underlies why we want to understand the fundamental mechanisms that give rise to these population behaviors. And we have a really strong history that we can rely on because of the cellular biochemistry that has been done over many decades in the context of T-cell biology. Pardon me. And what you can see here is the biochemical signaling cascade that has been developed such that we can see that TCR pMHC binding, so between the green receptor and cyan ligand, kicks off a series of intracellular events, largely through phosphorylation cascades, that creates signaling assemblies templated like on the molecule lat that you can see shown in red, and eventually leading to downstream signaling and translocation of transcription factors like ERK and NFAT. Furthermore, there's really important information in the physical arrangement here at this junction between the T cell and the antigen-presenting cell because of the changes in the landscape that occur when these ligation events are encountered, because you can see on the right-hand side of the screen that larger molecules like CD45, which is a phosphatase, and adhesion molecules like the interaction between LFA and ICAM are actually excluded from this space based on their size. So again, the idea of the membrane as a reaction landscape becomes really important as we think about the underlying activities that are setting the decision-making criteria. And what I mean by that is for a long time, it's been uncertain what actually sets the threshold for a T cell to activate. And so we started trying to understand this problem using normal T cells, even before thinking about engineered ones. So I'll briefly discuss how we translate these very complex problems that involve three-dimensional junctions between T cells and target cells to approximate just the intermembrane space and gain as much high-resolution information as we can. So on the left, you saw a very short video of a T cell sampling the surface of a tumor cell. In the middle, you see the schematic of the typical junction formed between a T cell and an antigen-presenting cell. What we can do is we can approximate this by replacing the antigen-presenting cell with a supported lipid bilayer that is glass-supported. And the benefit of this is that by having a mobile bilayer that we can derivatize with any molecules that we would like and at any densities we would like, we can then create a scenario where we can gain a lot of information through direct visualization using microscopy of events that are occurring at that interface. And so the video that's playing on the right is showing you the footprint or the contact area between a living T cell and the supported lipid bilayer shown in the RICM channel. And then the ICAM-YFP channel that you see in the lower left shows you the adhesion that I had mentioned very briefly in the prior slide. So you can see that this contact area is rich in interactions that are keeping the T cell on the bilayer. And I'll talk about the binding events that are shown on the right in a couple of slides. So by taking this reconstitution approach where we have the interface between a living cell and this synthetic antigen-presenting cell, we now give ourselves an opportunity to take advantage of high spatiotemporal resolution imaging, namely in a TURF configuration. And what you can see in this slide is that the freely mobile Brownian diffusion of individual pMHC molecules, in orange, occur on this synthetic APC, but upon interaction and bona fide binding with the T cell receptors of a living cell, the mobility that results from the bound complex between T cell receptors and pMHC leads to slowed mobility. And that change in mobility is something we can directly measure as a reflection of the binding events that are occurring. Push to audience. Okay. So on this movie, what you can see is individually labeled peptides, pMHCs, that are diffusing freely on the bilayer in the upper left, and you can see that they undergo normal Brownian diffusion. But in the presence of a cell, which is shown toward the upper right and outlined in cyan, is that in the presence of a cell, there are in fact two populations, a fast-moving population and a slowly diffusing population, which is in fact the bound events. So we can then select for the bound receptors simply by elongating the exposure time. And therefore, as you see in the lower right, things that are moving quickly are blurred, and only those binding events that are occurring on this timescale are clearly resolved. So by taking advantage of that information, we can now individually watch the binding events that are encountered by a single living T cell. And this is important because it gives us information about when a signaling cascade may be kicked off. So I revisit this cartoon of the biochemistry of TCR signaling to remind you that once you have bound receptors, a number of downstream events are possible and a cellular response is possible. But in order to try and answer the fundamental question of what activates a T cell, we tried to simplify this idea using an analog to signal processing. So we take the idea that this could be an impulse response function. And by impulses, I mean discrete pieces of information that go into the system. So here our system is going to be the cell. And these impulses can be modulated either in amplitude or in frequency. That is, how strong an interaction might be, a binding event, or how frequently they might be encountered. And this black box is in fact all of the cellular biochemistry that's happening in the cell that is so complex and difficult to measure, but critical to the transformation process that the cell will then act on to create a response, or the input-output response function. But what's really beautiful about this response is we can imagine that by measuring the individual binding events that a T cell experiences, and without measuring all of the intracellular complex biochemistry, if we can determine that the cell did something, made a decision, and in this case we take the binary response of translocation of a transcription factor NFAT into this, we now can determine the transformation through extraction rather than through direct observation. So it's easiest to understand this by actually seeing it in the context of a movie, and what you'll see in the movie I will play is this RICM channel, which again shows us membrane topography. So we will see living T cells come and land on the bilayer. And in the middle channel, the pMHC ATO channel, this is where the inputs are being measured. So each of the binding events that the T cell experiences. And then finally on the right-hand channel, in the NFAT, we can see the translocation of the transcription factor NFAT into the nucleus. And as I mentioned, this is a binary decision. So once the transcription factor goes into the nucleus, that cell is committed to activating. And so we'll watch this process in the movie, and then I'll make a couple comments after that. So here we see the cell land, and it spreads and forms an interface that we see here in RICM. We can see the circled binding events in red. And then over time, we'll notice that the NFAT goes from the cytoplasm into the nucleus, and that translocation demonstrates that this cell has activated. It may be a little easier to look at these data in a more static sense. So here's a montage of that same movie. And I'll point out a couple of really important features. The first is that we can directly visualize the process of cell landing that precedes binding events and the decision-making process, thereby having the complete history of information that this cell experienced. So we know the moment it started interpreting information from its local environment, what those information looked like in terms of single molecule binding events, and ultimately whether or not the cell activated. So all of these pieces of information allow us to have a timeline as well as spatial information about all of the impulses that went in as well as the response. And one could imagine that by knowing the impulses and the response, we can gather information about the transformation. So here we can see all of that information collapsed into a simple 3D movie where the binding events that are being accumulated, so the binding events shown as magenta circles in the movie that's playing, create information in the form of three-dimensional trajectories in time. So each of the lines that you see in this, what we call spaghetti plot, each of the noodles, if you will, shows you where the bound events occurred in time and in space for this particular cell. And you can also see in the topmost plane, shown in green, the activating information in the form of the NFAT translocation event. So I'll go ahead and play that one more time. So given this time evolution, we have all kinds of information about what this cell experienced. We can take these kinds of data and then try to understand what led to making the activating decision. And remarkably, the simplest hypotheses fail. So the simplest hypotheses would be that the T cell counted the number of binding events it experienced, or that it summed up the total time of binding that it experienced, and that integrated binding time set the activation threshold. In fact, neither of these is the case, and rather what we found in the course of this work is that T cells are paying attention to both where and when binding events occur. So keep in mind that more often than not, binding events are very short because real agonist ligands have low affinities that are almost indistinguishable from self-ligands. And so in fact, what you can see in the schematic on the left is if you were to imagine one bound trajectory shown in blue that is experienced for some amount of time, and then it disengages, and another binding event shown in red that over some sort of temporal window follows the blue event, and is also somewhat nearby in space, what we call the spatial neighborhood. In fact, what appears to happen is the T cell artificially stitches these two events together and interprets it as one very long binding event. And from a biochemical perspective, there's a good intuition here that the longer the binding event, the greater the amplitude of signal, because the longer time there is for biochemical reactions to occur. And so quite remarkably, we were able to show that in fact what a T cell does in making an activating decision is use these multiply correlated tracks. So what you see here in this simplified schematic is the idea that shown in gray boxes are the amplitudes of signal. In the context of non-activating cells, we can see that short binding events lead to low amplitude or short, small amplitude signals, whereas long binding events, or these binding events that are spatio-temporally correlated, as you see on the far right, can sustain greater signal and lead to activation. So again, I want to emphasize a couple of features about this impulse response assay. The first is that we can see both activating and non-activating cells, and this is really important because it allows us to distinguish threshold activities for those cells that do make a decision, say, to activate, and those that do not. And then finally, even though there is this spatiotemporal correlation, it's quite interesting that the scales of these correlations are larger than we might immediately predict. So events that are within about 2 microns in space and 30 seconds in time can be stitched together. And I don't have time to talk about the underlying machinery, but we also have newer data, hopefully in an upcoming publication, that show that this LAT molecule that I briefly mentioned earlier may be mediating this spatiotemporal correlation. But ultimately what I want to go on to is the discussion of what does this mean in the context of chimeric antigen T cells. And so this is the work that we've been expanding since I moved to Purdue, where you can imagine that now by knowing that T cells are capable of spatiotemporal correlation, we could imagine the same might be true for engineered T cells, but if it's not, we would probably want to know this in order to better engineer robust responses moving forward. So the idea here is that by having an artificially engineered receptor that can recognize a tumor cell, you could create an activation cascade within a T cell from a patient that then leads to one of two types of activation, either mobilizing adaptive responses from other cells or perhaps directly killing the target tumor cell. And so we still ask the underlying question, what are the molecular features that control this activation? And it'd be tempting to think that these chimeric antigen receptor T cells signal exactly like a TCR, but in fact we have plenty of evidence that they are quite different. The first is that canonical structure that I talked about that allows for concentration of bound TCRs at the center and other binding events of larger physical dimension like adhesion that creates this structure called the immunological synapse that you see in the overlay, where there are very tight TCRs in the center and then ICAM-bound adhesion complexes in red outside there. There's plenty of evidence that in a chimeric antigen receptor T cell context, this very well-known structure of the immunological synapse does not exist. And so already there are good reasons to think that CARs signal differently and that we need to better understand how they are signaling. So in partnership with Phil Lau's lab here at Purdue, we've been taking advantage of an engineering strategy that Phil's lab has developed. And so this takes advantage of a slightly different strategy that is called a uniCAR. So rather than a CAR alone, this universal strategy is based on having CARs that recognize a single molecule, FITC, which is not one that you would see physiologically, but that by having that FITC molecule on a bridge, here we show FITC folate, then the folate, or the folic acid, binds to the tumor itself and bridges between the tumor and the CAR. This carries the benefit that you could then have a single population of CARs that could be targeted to many different cancers simply by modifying the bridge. The other convenient thing is that since FITC is a fluorescent molecule, it can be used in laser-guided surgery because, as you can see toward the upper right, by having the FITC folate within a tumor, you can see it glow and light up, which may also help in tumor resections. But with Phil's lab, what we're trying to do is to understand how a uniCAR a universal CAR actually activates. So what my student has been doing is creating bilayer systems that have the folate receptor on it displayed at different densities, and then using the bridging molecule FITC folate and the anti-FITC CAR to better understand the activation thresholds in several different parameter spaces, the effects of affinity of both the CAR itself and of the bridge, the densities of the molecules, which could be the densities of the CAR receptors on the surface, the density of the folate receptor, and then copy numbers and clustering. So of course, as I mentioned very early on, small numbers of signaling molecules may have large effects. And so we're trying to understand, using this reconstitution approach, how all of these different parameters fit into the activation decision for a CAR. And you can see our impulse response function exemplified here, where again, we assume that much of the biochemical signaling is as it normally would be in a T-cell receptor system, but have good reason to think that there may be additional pathways that are in play, because unlike a TCR, a chimeric antigen receptor T cell is only a single pass transmembrane receptor, so it is fundamentally quite different than a TCR. But our impulses of binding events between the CAR and the target folate molecule on the folate receptor can then lead to activation decisions in at least two forms that we're thinking of right now. One is transcription factor translocation, as we've seen before in other T-cell contexts, as well as the secretion of lytic molecules like perforin and granzyme. So these are actually the prototypical molecules secreted to actually kill target tumor cells. And we're working to better understand this impulse response function in the context of breast cancer. Another development that Kevin has been working on is to try and increase the sampling of the events that we are seeing. So one challenge that we faced in the early development of the impulse response function is the need to capture the landing and spreading of T cells. And I mentioned how powerful this information is because it sets the full timeline for that T cell. But of course, it can be difficult to capture this event. So Kevin is also working on strategies to increase the number of events that we can visualize simultaneously through larger fields of view. And so you here can see T cells coming in and landing on a bilayer, and you can beautifully catch at least a few, maybe up to 10 or more binding events, landing and spreading events for T cells at the same time. As we try to directly look for single molecule CAR binding events, as you'll see in the next two movies, we can watch these very dynamic CAR T cells using our reflection interference contrast channel, and we're getting very close to having really beautiful data of single molecule binding. So I'll play that in the next two movies. So here you can see a CAR on a bilayer, and you can see its membrane fluctuations as it samples the information around it. Just really beautiful T-cell interactions with the SLB. And in this video, you can start to see binding events as these really bright spots that are coalescing underneath that T-cell that we had just been visualizing. And so clear evidence that these binding events are being accumulated. So moving forward, we are now able to see these binding events between the folate receptor and the CARs, and where this is occurring in time and space. And this is giving us the ability to start looking at these impulse response functions and complex signaling circuits in search of the key thresholds for activation so that we can better design CARs and these signaling circuits to maximize killing efficiency. But you might ask the question, how does this then translate back to the three-dimensional context? Because of course reconstitution does lack some of the information that would occur at a true cell-cell junction. So in parallel, we are also developing systems that allow us to control this 3D junction in such a way that we can use living tumor cells and living CARs, where we now know the densities of receptors on the surface of the tumor and can look at the killing efficiency by the CARs directly, thereby testing some of the theories and models that come out of our reconstitution methods. So hopefully a lot of exciting things are on the horizon. My student has been doing an absolutely fantastic job building up these systems, and we really are poised to measure the impulse response function across many orders of magnitude for many cells to better understand how these CARs are being activated and what the threshold criteria are. So with that, I'll thank the members of my group. So this is the current group, Kevin being my main student, Ari, another graduate student through a collaboration, and our 3 undergrads. And I often tease my students that one of the skills we have to learn in science is about juggling, and I'm really pleased that they're all tackling this challenge as difficult as it can be at times, and really doing a fantastic job trying to become really great interdisciplinary scientists. And finally, it's really important to also acknowledge the wonderful collaborators and mentors that I've had and continue to work with. So Jay Groves at UC Berkeley, I was with him when we developed the impulse response function, and now we're getting to apply it here at Purdue with Phil Lau, and we have a host of other really fantastic collaborators and many fun and exciting things on the horizon. So thank you all for your attention. Thanks again to the organizers for the efforts and the invitation, and I'll look forward to future interactions and questions.